My name is Kenichi Okuda, working at UNCCF Center. Today, I'm going to talk about cell type and region-specific Cftr and secretory mucin expression in human airways. There are no COIs to disclose related to this presentation. Airway mucociliary clearance, MCC, is a critical innate defense mechanism to protect the lung from potentially harmful particles and pathogens in inhaled air. The MCC is composed of mainly three airway epithelial cellular activities. First activity is mucin secretion into mucus layer. Airway surface is covered by mucus layer to trap the inhaled particles and pathogens. Secretory mucins are gel-forming macromolecules, and they provide airway mucus with viscosity. The second activity is trans-epithelial ion transport, which controls airway surface hydration. Key players in this activity are Cftr and ENA. The third activity is coordinated ciliary beading, which is necessary to efficiently transport mucus towards mouth to get rid of pathogens from the lung. This is a 2-gel model of airway surface liquid in normal airways. Mucus concentration in mucus layer is regulated by well-balanced fluid and solid components like mucins. In CF, Cftr dysfunction causes reduced fluid secretion, which causes mucus hyperconcentration, depletion of PCL, and eventually MCC dysfunction. Indeed, higher concentration of mucin is identified in CF sputum and BARF compared to normal samples. Also, in vitro culture models demonstrate the reduced PCL height in CF airway epithelial cell cultures. Overall, this model indicates that mucin fluid secretion into airway lumen is critically important for normal MCC function. Here, I'd like to talk about structural difference between large and small airways. Human conducting airways typically consist of 23 times branched airways. Small airways are defined as airways with diameter less than 2 mm, which are also called bronchioles. Presence of cartilage and submucosal glands are a unique feature to large airways. The histology images of epithelial cells shown here demonstrate morphologically different cell types present in different airway regions. Despite these known differences between large and small airways, how are mucin fluid secretions regulated in different airway regions is unknown. Particularly as small airways are the most difficult region to study due to limited access, the MCC mechanism in small airways are largely understudied. Indeed, mucus obstruction appears to occur in small airways in CF. Micro-CT data exhibit mucus obstruction starts at 6 to 7 generation bronchi to distal airways. Also, lung particle clearance data show reduced particle clearance, particularly in small airways in CF. Given these data, it is important to understand region-specific MCC systems, particularly in small airways. Based on these background, we try to characterize measure airway secretory mucin, MUC5B, 5AC, and Cftr expression throughout the human airway, particularly focusing on their regional distribution and expressing cell types. Firstly, we try to understand differential distribution of major airway secretory mucins, MUC5AC and 5B. Between these two secretory mucins, there are reported differential features. Firstly, the source of these two mucins are reported to be different. While MUC5B is reported to be mainly produced by submucosal glands, MUC5AC is predominantly secreted by goblet cells in superficial epithelium. Secondly, MUC5B is detected 10 times more than MUC5AC in normal human airway secretions. This MUC5B-5AC ratio may be altered in disease. Thirdly, in mice, MUC5B is required for normal MCC and survival, but knockout of MUC5AC gene did not alter either MCC or survival. So these data suggest MUC5B may be basal mucin that maintain MCC, whereas MUC5AC may be reactive mucin in stress condition. In order to understand the differential properties of 5AC and 5B in human airways, Characterizing 5B, 5AC distribution in healthy human airways is important. So we localize MUC5 and 5AC mRNA and protein in human airway tree by RNA-seq hybridization and immunohistochemistry and quantified those signals. While MUC5B is exclusively expressed in submucosal glands, both 5B and 5AC are abundantly present in the large airway superficial epithelium, like trachea and segmental bronchi. In bronchioles, which are small airways at the diameter less than 2 millimeters, while MUC5B is still present, MUC5AC is not detected. Finally, in terminal respiratory bronchioles, neither 5B nor 5AC was present. We tried to validate those morphometric findings by measuring absolute number of 5B, 5AC transcripts in airway epithelial cells isolated from freshly excised different regions of airway tissues using droplet digital PCR. Consistent with morphometric data, while 5B and 5AC transcripts are present in large airways like trachea and bronchi, MUC5B is predominantly identified in bronchioles. So these data clearly suggest MUC5B dominates small airway regions. So to extend our 5B and 5AC regional distribution findings, we estimated total mucin expression in each airway region to understand which airway regions are the predominant site for MUC5B and MUC5AC expression in the whole lung. As shown here, total surface area of small airways is exponentially greater than that of large airways in the lung, reflecting an exponential increase in number of airways in small airways. Since MUC5B is expressed in small airways, our calculation reveals the small airway region as the predominant site for MUC5B expression. In contrast, since MUC5AC is absent in small airways, the predominant site for MUC5AC expression is large airways. Despite the exponentially greater surface area in small airways. So based on this estimation, MUC5B is predicted to be the predominant mucin in human airway mucus. So next we looked at cell types expressing MUC5B and MUC5AC in different regions of airway in association with airway secretory club cell marker CCSB. The gene name is SGGB1. In normal human airways, we found that both 5B and 5AC are expressed in SGGB1 positive secretory cells throughout the airway. However, expressed mucins are region-specific, consistent with 5AC or 5B regional distribution. I mean, while 5AC, both 5B and 5AC are co-localized in CCSB positive cells in large airways, like primary bronchi, CCSB positive cells express only MUC5B in the distal airways. In the terminal bronchioles, CCSB positive cells are still present, but they don't express either 5B or 5AC. So interestingly, these data suggest CCSB positive secretory cells can be classified into region-specific subsets. So we also looked at regional 5AC, 5B distribution in CF airways. Although both 5B and 5AC are reported to increase in CF sputum, MUC5B and 5AC distributions observed in normal subjects are preserved in CF airways. I mean, MUC5AC expression is mainly upregulated in larger proximal airways, whereas increase in MUC5B is more significant in small airways. So interestingly, in the terminal bronchioles, MUC5B expression appears to be extended to this region in CF. So this MUC5B extension may be relevant to small airway disease in CF. So here I'd like to summarize our findings about 5AC and 5B distribution in the normal human airways. Our data demonstrate that small airway is the major site of MUC5B production, whereas proximal airway is the predominant site for MUC5AC production in the lung. So CCSB positive secretory club cells are responsible for 5B and 5AC production with region-specific manner. Neither secretory mucins are produced in very distal terminal bronchioles. So now let's move on to the CFTR. Historically, multiple airway epithelial cell types have been reported to express CFTR in human airways. Dr. Engelhardt reported non-ciliated cells express CFTR in bronchioles and submucosal gland ducts in 1990. Later, immunohistochemistry-based approach identified CFTR protein expression in apical plasma membranes of ciliated cells, as well as rare hot cells in bronchial surface. Then, recent single-cell RNA sequence studies identified CFTR-rich pulmonary ionocytes in human large airways and submucosal gland ducts. However, question as to what cell types express CFTR has not been fully addressed. So we analyzed CFTR expressing cell types in large and small airways in two single-cell RNA sequence cohorts using different sample collection methods and single-cell RNA sequence platforms. You can see the dense CFTR positive cell clusters on the right UMAP in the location corresponding to secretory, suprabasal, and basal cell clusters, in addition to the ionocyte cluster. So we estimated each cell type's contribution to total CFTR expression by combining average CFTR expression levels per cell and the frequency of CFTR positive cells in each cell type. The results of the two cohorts matched pretty well, and then demonstrate the secretory cells are the most predominant cell type expressing CFTR in both large and small areas. The secretory cells are this blue part in this pie chart. Interestingly, the contribution of secretory cell type to CFTR expression was greater in small areas than large areas. In contrast, we also noted ionocyte contribution is greater in large areas compared to small areas. Since our single-cell RNA sequence data revealed secretory cells are the cell type most contributing to CFTR expression in the airways, we looked at the relationship between CFTR and mucin expression on our single-cell data. Firstly, in total large and small airway epithelial cells, about 30% of cells express MUC5B and or 5ASC in both regions. Next, when we looked at these CFTR positive cells, about 40% of large, and then more strikingly, about 70% of small airway epithelial CFTR positive cells also express MUC5B and or 5ASC. So this data suggests airway secretory cells express both secretory mucin and CFTR in one cell, and then that is more significant in small airway regions. So as our single-cell RNA sequence data demonstrate a greater contribution of ionocytes to CFTR expression in large airways, we looked at regional distribution of CFTR and ionocytes throughout the normal and CF airways. While CFTR expression is constant from large to small airways, ionocyte specific marker FOXI1 positive cell is decreased in the small airways. This CFTR and ionocyte distribution pattern is also observed in CF airways. And then this data may imply the importance of non-ionocytes contribution to CFTR expression and function in small airway regions. We posit that may be a secretory cell. Our single-cell RNA sequence data provided powerful information about CFTR expressing cell types. However, our single-cell RNA sequence studies identified CFTR expression in only 3 to 6% of airway epithelial cells. This low frequency of CFTR positive cells raises a question as to CFTR expression in such a small fraction of epithelial cell is sufficient to hydrate airway mucus that covers all over the airway surface. Also, drop-off is a well-known technical limitation in the single-cell RNA sequence technology to detect low-abundant gene expression. So we suspected single-cell RNA sequence may not be sensitive enough to detect all the CFTR expression. So then to figure out potential sensitivity issue in single-cell RNA sequence, we try to quantify CFTR gene expression in each major airway epithelial cell type with more sensitive method. Firstly, we generated single-cell site spin from large and small airway epithelial cells and performed RNA in situ hybridization to look at CFTR mRNA at single-cell levels. So our quantification revealed CFTR mRNA expression in 55% in large and 39% in small epithelial cells, which is about 10 times more than single-cell RNA sequence data. Further, we co-localized CFTR to measure airway epithelial cell type markers. The results reveal that majority of secretory cells, HGB1 positive secretory cells, express CFTR with relatively higher intensity compared to other cell types. Although ionocytes are rare, most of them express CFTR with the highest intensity. The frequency and intensity of CFTR expression is least in ciliated cells. Further, we utilize single-cell qRT-PCR to quantify CFTR expression in measured airway epithelial cell type at single-cell level. So we perform a single-cell-based multiplex qRT-PCR. After QC, 146 cells were classified into secretory, ciliated, basal, or other cell types depending on marker expression. We identified CFTR expression in 70% of epithelial cells studied, which is more consistent with single-cell RNA in situ hybridization data. Importantly, single-cell qRT-PCR identified CFTR expression in 80% of secretory cells, which is again consistent with single-cell RNA in situ hybridization data. So CFTR expression was not identified in ciliated cells in this dataset. Importantly, these two more sensitive methods still demonstrated robust CFTR expression in secretory cells. So based on all our CFTR expression analysis, finally we transferred to functional study to test whether wild-type CFTR transduction specific to secretory cells can restore CFTR function in CF airway epithelia. So we designed lentiviral vector carrying rat-CSSP promoter driving wild-type CFTR or GFP as a control. So we tested these lentiviral vectors in CF large and small epithelial cell cultures. Both wild-type and mutated CFTR protein were identified in rat-CSSP CFTR transduced cells, whereas no wild-type CFTR protein was detected in non-infection or GFP control cells. Whole mount immunohistochemistry reasonably localized CFTR protein to CSSP positive non-ciliated secretory cells. And most importantly, CFTR function is actually restored in rat-CSSP CFTR transduced cells, but this restoration was not observed in controls. These data suggest that secretory cells are competent to restore CFTR function in CF cells, supporting our hypothesis that the secretory cells can be the major cell type, normally expressing CFTR. So in conclusion, CFTR is expressed in multiple cell types throughout the human conducting airways. Among airway CFTR expressing cell types, secretory cells dominate CFTR expression in superficial epithelia, particularly in small airways. The secretory cells are competent to restore CFTR function and could be a potential target for cell-target molecular therapy for CF. So we studied region and cell type specific expression of major airway secretory mucins, MUC5A, C5e, and CFTR. All of these are critical components to regulate airway mucus concentration and MCC. Our data may propose a new paradigm that secretory cells play a central role in maintaining airway mucus homeostasis by regulating mucin and fluid secretion in one cell, particularly in small airways. I greatly appreciate all the help of my mentors and collaborators inside and outside the UNC shown here. I also greatly appreciate all the support from my sponsors. Thank you so much for your attention. I will be happy to take any questions.
I would like to take a few moments to try to review with you the structure and function of the large and small airways, especially the elements of fluid and electrolyte transport. So if you will, let me begin by giving an overview, a quick overview of the large and small airways, or what we're talking about when we say large and small airways. Basically, the large airways are the bronchi, extending, including the trachea and extending through the main bronchus and the segmental bronchus with continuous bifurcations and continually getting smaller at each bifurcation until we reach the region of the bronchioles, which are very small airways that also continue bifurcating into smaller branches, forming the terminal bronchioles, and then at last the respiratory bronchioles, which are the only airways that really participate in gas exchange. So small airways, bronchioles, large airways, bronchi. If we take a cross-section view of a large airway, we see that the lumen is covered uniformly by pseudostratified ciliated epithelia with goblet cells in between. This cellular arrangement allows that the cilia can propel fluids on their surface, which may contain debris and other discarded material, so that it doesn't accumulate in the lung and perpetuate infections or inflammation. The goblet cells form to provide mucus for this fluid so that the debris can be easily trapped. So if we move now to the small airways, that is the bronchioles, we see that the luminal surface is distinctly different in that it is highly invaginated. That is, it has many villi and pleats, or pleats and folds in the villi. This structure, of course, accommodates the ability of this small airway to expand and contract without changing the surface area of the lumen of the bronchiole. So this thing will expand as we inhale and will collapse as we exhale. Briefly to the left here, we see a normal condensed bronchiole with this patent airway contrasted to a CF airway, which has been filled with mucus and is plugged. So the villi are not as extensively expanded, extensively collapsed as in the normal airway. And of course, this airway serves no purpose in conducting air to the alveoli. So quickly, if we compare the major properties of the bronchi and the bronchioles, first we say that they are both lined with ciliated columnar epithelial cells. The bronchi are characterized, the epithelia of the bronchi are characterized as being pseudostratified, but they are both luminal surfaces are highly ciliated. In the bronchus, we have cartilage rings and plates embedded in the submucosa, which of course maintains the patency of these larger airways and keeps them from collapsing. Beneath that, or among the cartilage and beneath the epithelia in the submucosa, we have mucosal glands, which provide fluid and some mucus to the airway surface in the bronchi. On the other hand, in the bronchiole, we have no cartilage. There's no rings or plates in the bronchioles, and the bronchiole has no mucosal glands. So the surface epithelia has to do all the absorbing and secreting of the bronchiolar fluid, airway fluid. So now let's take a look at the components that are responsible for maintaining and managing the airway surface fluid in the large and small airways. We can get a view of this by the effects of our inhibitors and agonists on short circuit current when these tissues are examined externally. That is, if we mount these tissues so that we can measure the short circuit current, the current activity will tell us whether we have an effect of the inhibitor or the agonist and let us make comparisons. So here we see, if we apply amiloride as an inhibitor And as you probably know, amiloride is a very selective inhibitor of the sodium channel, and it prevents sodium absorption. So if we apply amiloride to a non-CF lung and look at the effects on the large airway tissue versus the small airway tissue, we see that it affects both almost equally. There's significant inhibition of the current, and it inhibits them almost equally. In the CF tissue, it's about the same. It inhibits ENaC in the large airways about almost to the same extent as it does in the small airways, and there is no difference between its effect in normal tissue or non-CF tissue versus CF tissue. Now, if we move over and look at DIDS as an inhibitor of the NACFC, that is a sodium potassium 2-chloride cotransporter, again, if we apply it to the non-CF large and small airway, we see virtually no difference in the inhibition. If we apply it to the CF tissue, the large and small airway tissue, again, we see virtually no difference between the two, and the effect in both the non-CF tissue and the CF airways is about the same. The inhibitory effect is about the same in both sources of these tissue. However, if we now look at an inhibitor that is relatively specific for Cftr, that is the protein that's affected in cystic fibrosis, we see that there is a dramatic effect of this inhibitor and a dramatic difference between its effect on large airways and small airways in non-CF tissue. So that perhaps is somewhat enlightening. At the same time, if we look to the effect of GlyH on CF, large and small airways, we see virtually no difference in the effect, and the effect is really quite small. Now, if we look at an agonist, that is the agonist of forskolin plus IBMX in order to increase adenyl cyclase activity to produce more cyclic AMP, which stimulates CFTR, we see that in the non-CF airway, it has a much smaller stimulatory effect on the large airway than it does in the small airway, that is, for non-CF tissue. In contrast, it has virtually no ability to stimulate the CF tissue in either the large or the small airways. So that should not be so surprising since it is CFTR that is defective in the CF tissues. So that characterizes our the major transport components of our bronchiolar epithelium. So we can't get by without taking up bicarbonate, because it is a major constituent and extremely important and critical to the pathogenesis, I believe, of cystic fibrosis, as much, if not more, than anything else. And here, if we look at the human bronchiolar tissue, we find that forskolin has a dramatic effect on stimulating the short circuit current in these cells, and GlyH, the specific inhibitor of CFTR, has a dramatic effect of inhibiting the stimulated secretion. Just as a side note, if we look at the calcium-regulated chloride channel, which is stimulated by UTP, it is also present in the bronchiolar tissue, and that channel can be inhibited by diflumic acid, and we see dramatic responses to both stimulation and inhibition in this graph. So the location of our transport inhibitors, of our transport mechanisms. So ENaC, the epithelial sodium channel. When we look for it immunohistochemically, we find it located almost exclusively on the apical surface of the folds of the villi of the bronchioles, as you see it displayed very nicely around this circumference. It does not appear to bind, or to be present, that is, in the pleats of the villi. So the absorptive tissue appears to be here on the surface of the folds. And now, if we look for CFTR, we see that it's associated with the apical membrane. stains also on the folds of the villi, but also in the pleats of the villi, so that it looks as though the folds and the pleats both contain CFTR to participate in both secretion and absorption. And then if we look at NKCC to indicate secretion, in contrast to the localization of ENaC, which was in the SARC2 cells, NKCC is associated with secretory cells, and we find it localized here in the base of the pleats of the villi to support secretion. So how do we make sense of all this? How do we put this together? Oh, I almost forgot bicarbonate. If we now localize bicarbonate as an important component for secretion in the airway fluids, we look for the sodium bicarbonate co-transporter and the sodium bicarbonate exchanger, and in the bronchiolar pleats, we find that it is localized in the base of the villi. Very little expression in the folds of the villi. So putting all this together, if we can be, I guess, somewhat speculative and suggest from this model of a villi with the pleat here and the folds here, airway surface coating the whole thing. And what we would suggest is that the base of the pleat is responsible for secreting the airway surface fluid, and that the folds up here are responsible for absorbing the airway surface fluid. So in the villi, we secrete the fluid, and in the folds, we absorb the fluid. In the pleats, we secrete bicarbonate, and the bicarbonate is very important to provide the ambience or the environment for secreting mucins. When the mucins are secreted in the presence of bicarbonate, they explode and are released into the ASF so that they can be transported as large macromolecules and not as condensed, compacted granules. So if we put all this into motion, we see something like this. That is a recirculation of the airway surface fluid moving as it's secreted from the villar portion to the surface or through the surface of the folded portion of the villi. So with this arrangement, we might predict that this makes it very difficult for the airway to ever be completely dry because there is no absorption deep in the pleat, and it can never be completely flooded, or shouldn't be unless it's a pathological condition, normally flooded, because there is absorption of the secreted fluid in the fold of the villi. So we have this recirculating fluid that keeps the airway nice and wet without allowing it to flood and drown us. So what do we got? Well, I would be so base as to say we have a washing machine in our breathing machine. So by what we just saw, the villi and the folds will move, contract, and expand according to in concert with our respiratory cycle. That is, as we inhale, we expand, and as we exhale, we contract. Inhale, expand, exhale, contract. Inhale, expand, exhale, contract. So we can envision then that this airway surface fluid, in the process of inhaling and exhaling and contraction and expansion, is being agitated. It's being moved or stirred, if you will, so that it doesn't become stuck and accumulated in a particular region. It can be maintained mobile and moved more easily across the luminal surface. So, in conclusion, I think we might say that CFTR is crucial for the formation of airway surface fluid in small airways. The airway surface fluid is critical to small airway hygiene and clearance. and the loss of CFTR function impedes small airway clearance, and therefore the small airways are most vulnerable in cystic fibrosis and probably in normal bronchioles as well. So I would like to thank you very much, along with my team. I hope you remember them, and will continue to remember my good friend Nate, my lovely lady Cora, my good friend Bicarby, and my good friend K-Boy. So thank you, and I hope to see you all very soon. It's been a long time. Thank you.
Hello, it's a real pleasure to be able to even virtually present this talk on non-CFTR ion channels as an alternative approach to addressing mucociliary dysfunction, specifically in cystic fibrosis airway disease. And I wanted to thank the organizers for inviting me. I'm Matthias Salate. I'm at the University of Kansas Medical Center, and I'm specifically interested in one of these ion channels, and I will talk much more about that in a few minutes. First, there is one relevant disclosure to announce. This is an advisory board for Airway Pharmaceuticals, but that should not influence the talk that I'm presenting today. So what are the major ion channels in airway epithelia? And I'm concentrating here really on the apical membrane. It's obvious the CFTR, but in cystic fibrosis, at least without effective modulators, this channel is dysfunctional. And alternatively, there is TMEM16A. That's a calcium-activated chloride conductance that can also conduct bicarbonate, SLC A26A9, and then a potassium channel. We'll talk much more about that. All of those channels really provide a driving force for water to hydrate the airways and therefore hydrate mucus, and thereby decreasing mucus concentrations for appropriate mucociliary clearance. On the other hand, there is ENaC, that is a sodium absorber, and is known to dehydrate the airways by pulling water away, thereby increase mucus concentration and causing mucociliary dysfunction in the appropriate settings. So where are these channels actually located? These channels, meaning in what cells are they located? I already indicated that these are at the apical membrane. Well, this has been a debate, and I don't have really time to go into this debate today, but RNA-seq data has indicated very interesting challenges to previous beliefs. There is expression in ionocytes specifically, and some of these channels more in secretory cells than ciliated cells. And I think the judgment of all of this is still going on, and the debate can be at a different conference. So let's focus first on the ENaC, the sodium absorber that dehydrates the airway epithelium and can cause mucociliary dysfunction. It's a complicated channel. Here you see there's three major subunits, alpha, beta, gamma, and they work in concert to absorb sodium. How do we know that ENaC is important for mucociliary clearance? Well, we know from old data from patients with a rare mutation that causes pseudo-hypoaldosteronism, that those patients with dysfunction in ENaC have a significantly increased mucociliary clearance, as indicated here. They clear radioaerosol inhaled much faster than normal people. Now, these are not cystic fibrosis patients, but we also know that some rare variants in ENaC subunits can actually provide longevity in cystic fibrosis and provide a non-progressive phenotype. Again, these are rare, but indicate that ENaC inhibition can actually help the lung disease in cystic fibrosis. It is not surprising, therefore, that a lot of companies have started their work in ENaC inhibition, and unfortunately, a lot of those trials were unfortunately negative. And you see that in red here, that a lot of those trials did not result in significant improvement in cystic fibrosis patients. Some others were paused or discontinued based on some toxicology studies. And again, the judgment on this is outstanding, and whether or not they come back will have to be determined. But why did so many therapies really fail here? One of the hypotheses is that many of those therapies were tested in sheep. This is a non-genetic model of cystic fibrosis airway disease. And the inhibition of ENaC in these sheep led to significant improvements in mucociliary function. However, when extrapolated from the sheep to the human, it seems that many of these medications were underdosed. And if they had been dosed higher, and that was probably prevented by toxicology studies, they could have had an effect. This is a hypothesis, and we will have to wait until additional studies are being presented where dosing has been increased, and then hopefully showing an improvement in mucociliary dysfunction in cystic fibrosis. An alternative channel is TMEM16A. Again, this is a channel that is calcium activated. It's part of the channels that provide the calcium activated chloride conductance. The activation is classically through ATP and the P2Y2 receptor, and therefore initial attempts of stimulating this channel as an alternative chloride channel and also bicarbonate channel came through analogs of UTP, the mucosal. However, unfortunately, this trial failed as well on these trials. The idea here is that ATP is actually elevated in cystic fibrosis airways, and therefore this cascade of signaling may have been downregulated, thereby not providing an avenue to really stimulate this channel. So potentiators are being actively sought, even though there are potential side effects as bronchoconstriction and mucus secretion. However, here, one of those potentiators, ETX001, in cystic fibrosis airway epithelial cells shows a significant increase in availability of water on the airway surface compared to vehicle, as seen here. And also in a sheep model, again, a non-genetic model of mucociliary dysfunction in sheep, with CFTR inhibitor inhalation and decrease in tracheal mucus velocity, there's a significant improvement of tracheal mucus velocity with the potentiator of TMEM16A. The same is true for whole lung clearance with aerosol inhalation, even though this is in a sheep that has not seen CFTR inhibition. So there is very good data to see whether we can move forward with clinical trials with TMEM16A potentiator. Now, the question was whether or not this causes mucus secretion that might be detrimental, and also bronchial constriction. These data here that I don't really want to discuss in full detail show that this particular potentiator does not increase mucus secretion and is not related to bronchoconstriction, ameliorating these concerns. The next alternative channel is SLC26A9. This is really part of the solid carrier family of anion transporters. It's mainly transporting chloride. However, the problem with SLC26A9 is that it is trafficked to the apical membrane with CFTR, and there are no known potentiators or correctors. So either it can be improved in function with CFTR correctors, or we will have to find new correctors for SLC26A9 in particular. However, we know that overexpression of SLC26A9, at least in the pancreatic duct, has shown improvement in pancreatic function, specifically as it delays CF-related diabetes. And we also know that certain SNPs of SLC26A9 in mutations responsive to Cftr, so these are not trafficking mutations, improve lung function as well. So there is more to come, hopefully, on SLC26A9, and this is also a promising avenue for therapy. Now let's focus on the channel that I really is close to my heart and that I really like. This is a potassium channel, the calcium-activated voltage-dependent potassium channel with large conductance, also known as PK. It's a complicated channel That's a tetramer opening up a channel here with the alpha subunit. Also has multiple beta subunits here in green. We know that in the airway, beta 2 and beta 4 are mainly expressed, but there is also an important gamma subunit here in purple. This is called LRRC26. There are other LRRCs that provide the same function, but this subunit is important because it allows the BK channel to open without a significant voltage change and thereby make it basically dependent on calcium activity rather than voltage change. So why is this important, and why would we be interested in a potassium current at the apical membrane? Well, when you think about how chloride actually exits the cell, the driving force needs to be provided, and there is good data that the driving force can be provided by basolateral potassium channels, but the apical channel can help as well. And this is depicted here in this model, where increasing potassium conductance, specifically in red here, clearly increases the current provided by chloride at the apical membrane in about almost twofold compared to no potassium conductance. This obviously relates to an increase in potassium concentration in the airway surface liquid, and in the here worst case quotation mark scenario, this is in the order of 30 to 35 millimolar. However, this is in agreement with measurements actually in the airway surface liquid, at least in mice, and therefore this model of increasing apical potassium conductance, easing the chloride exit at the apical membrane, seems important and relevant. This is shown here in now experimental setup where paxillin is used to inhibit the BK channel. And you can see that with paxillin, the calcium response in terms of airway surface liquid to UTP is completely abrogated. In addition, this is not only done by paxillin, but by a highly relevant cytokine related to worse outcome in cystic fibrosis airway disease, namely TGF-beta1. TGF-beta1 completely abrogates the response to UTP as well. And therefore, it seems that when you decrease the ability of the potassium channel to provide apical potassium exit, there is a dehydration of the airway epithelium. This is shown here in red. If you look at airway surface absorption without actually UTP, there's a clear increase in airway absorption. And interestingly, we tried losartan as an angiotensin receptor inhibitor that has anti-inflammatory capabilities and has been used in Marfan syndrome that is related to TGF-beta successfully in clinical trials to counteract the TGF-beta effects. We can ameliorate the ASL absorption. This is also reflected by an improvement in ciliary bead frequency, basically availability of water at the apical surface, and importantly, a decrease in the initial increase of mucus concentration by TGF-beta in these airway epithelial cells. The same is true for the BK channel. You see the BK channel activity in these cystic fibrosis cells is clearly decreased in the presence of TGF-beta, and it can be partially rescued by losartan. This is mediated by LRRC26, this important gamma subunit. You see that we decrease with TGF-beta the LRRC mRNA in these cells significantly, and losartan has a rescued effect. However, losartan must have some additional effects that we don't fully understand, because it has a very quick active effect after 5 minutes in these cells on the BK channel as well. And it improves BK channel activity in the presence of TGF-beta as well within 30 minutes. So this is not LRC mediated, but an additional effect on the BK channel. So these are cells. Is this relevant in any animal model? Before I go there, I want to show you the inflammation caused by TGF-beta, COX-2, IL-8, and IL-1 beta, a very important cytokine in cystic fibrosis airway disease. Their increases through TGF-beta are all ameliorated by losartan. Now, does it have any relevance to humans? Here are nasal scrapings depicted with neutrophil influx into the nasal airways and the expression of LRC26. You see there is an inverse correlation. Therefore, there is clear evidence that inflammation in humans is actually related to a decrease in LRC26 expression and therefore PK function. We went back to the sheep that I showed you already before. A sheep inhaled Cftr inhibitor and then human neutrophil elastase, an analogy to these human data. And you see there is an increase in TGF-beta relevance to the airway epithelial cell experiments, and also, again, mucus concentration associated with mucociliary dysfunction. And that is seen in this sheep experiment here. Cftr inhibition with TGF-beta leads to a prolonged mucociliary dysfunction for at least 24 hours, but it can be reversed almost completely with losartan. Now, this is an acute experiment. So we tried to create a more prolonged airway inflammation with Cftr inhibition and human neutrophil elastase inhalation for 3 days. That leads to a prolonged mucociliary dysfunction for at least another 48 hours. And you can see losartan is still able to reverse that mucociliary dysfunction. So we're very excited about that. And we wanted to do human studies, but these have been unfortunately delayed because of the COVID pandemic. Now, TGF-beta is also ameliorated in these sheep in these experiments with losartan, and so is the mucus concentration in analogy to the mucociliary dysfunction that I showed you in the slides before. So in summary for this part, there is inflammation that causes TGF-beta elevation in these at least airway epithelial cells and in the sheep, causing BK dysfunction. And we can, at least in the sheep model and the airway epithelial cell model, reverse that with losartan. Now, does that have any other indications in terms of disease? And we were specifically interested in cystic fibrosis-related diabetes because of the discovery that increasing the culture media in cystic fibrosis airway cells from a normal concentration of glucose from 5.5 to 12.5 is actually also decreasing PK function, decreasing LRC26 mRNA, and causing mucociliary dysfunction by increased absorption of airway surface liquid. This again has correlates with human beings. Nasal scrapings, again, of LRC26 expression are inversely related to multiple indicators of poor control of glucose by measurement of continuous glucose monitoring. So LRC26 goes down with worse control of cystic fibrosis-related diabetes mellitus. This has multiple consequences, and this is a busy slide. It increases inflammation with RAGE. It causes p38 phosphorylation. It causes changes in microRNAs, increasing in MEP9 and other inflammatory proteins, again, IL-1 beta. So clear indication that there are problems associated with airway inflammation and high glucose concentrations. So briefly summarizing here, concentrations of glucose lead to inflammation that again affect the BK channel and causing potentially mucociliary dysfunction through this channel directly related to the glucose concentration. And that may be an indication that cystic fibrosis related diabetes has actually additional effects rather than only increased infections risk. Now, can we do anything about that? And yes, we believe we can. You can see that metformin, an old medication for non-cystic fibrosis related diabetes, can actually rescue BK without affecting the glucose concentrations in these cells and rescuing the absorption. And we are actively working on understanding this effect much better. Does it have really any effect in the era of highly effective modulators? And so you see here, these are data from the CF registry that we analyzed for patients without or with CFRD being on ivacaftor for over 7 years. And you can see that actually the trajectory of lung function decline is worse in patients with CFRD. So this might or might not be related to the problem of BK dysfunction, but it indicates that we need additional therapies in this patient population. With this, I thank you very much for your attention. Here are all the people that helped in these multiple examinations, and I thank you for your attention.
Well, hi, my name is Sinead Weldon, and I'm based in the Wellcome Wilson Institute for Experimental Medicine in Queen's University, Belfast. I'd like to thank the organisers for giving me the opportunity to talk to you today about host defence mechanisms in the CF lung. And in this talk, I hope to shed some light on the topic and highlight some recent findings and raise some questions that I think we need to address going forward. I'd like to apologise in advance to all those researchers whose work I wasn't able to mention due to time constraints. So the following relationships exist related to this presentation. And I'm just going to start with just a very basic recap of what happens in the CF airways. So effective Cftr impairs mucociliary clearance and a range of other host defence mechanisms in the lung. CF airway inflammation is dominated by neutrophils, which produce reactive oxygen species, neutrophil extracellular traps, proteases such as neutrophil elastase, and a range of immunomodulatory mediators. Now, obviously, neutrophils have important roles in controlling infection, but despite their numbers, they're dysfunctional in the CF lung. Airway epithelial cells and a range of other immune cells, including lymphocytes and macrophages, also play a key role, including release of a range of molecules with immunomodulatory as well as antimicrobial activity, such as LL37, defensins, proteases, and antiproteases. And I'll talk about some of these in more detail later on. So as part of the recent omics explosion, single-cell RNA sequencing has shed light on the cellular heterogeneity that exists within the airway wall. So we've had the identification of CFTR-expressing ionocytes and secretory cells, as well as altered epithelial cell populations and differentiation trajectories in late-stage CF. But it's not just the epithelium. Single-cell RNA-seq sputum cells found a shift from alveolar macrophages in healthy control subjects to a predominance of recruited monocytes and neutrophils in CF. Now, as expected, neutrophils were the most prevalent in CF, but these cells exhibited a dominant immature pro-inflammatory archetype. So although both monocytes and neutrophils had pro-inflammatory features, these cells showed transcriptional evidence of abnormal phagocytic and cell survival programs, which will contribute significantly to impaired innate host defence and tallies with a lot of the ex vivo CF cell function studies to date. The neutrophils are typically considered terminally differentiated and ready for bacterial killing. However, in CF, there's lots of them, but they're not able to protect the airways. So new approaches and recent research have revealed that, like macrophages, neutrophils are plastic and can actually change their transcriptome and phenotype in response to the local environment. Upon recruitment to the CF airways, neutrophils reprogram to an aberrant phenotype termed GRIM that includes exocytosis of primary granules, which contain elastase, increased cellular metabolism, and reduced bacterial killing. Similarly, blood monocytes undergo significant adaptations upon recruitment. However, monocytes were not metabolically activated, but they did display decreased bactericidal activity. And we need to know more about this pathological activity of these cells to help us identify treatments that can address these defects and restore cellular function. So So we also now know from a range of observational studies that CF lung disease starts in the first few months of life with evidence of mucus plug-in, neutrophilic inflammation, changes in the lung microbiome, as well as structural lung damage. These changes can occur in the absence of respiratory symptoms, obvious respiratory symptoms, or infection, and they persist into childhood and adolescence. A number of cross-sectional and longitudinal studies have indicated that, like adult CF, there's a high variability in the severity of early CF lung disease. Now, although CF is a monogenic disorder, disease severity is influenced by a host of factors, including Cftr genotype, modifier genes, epigenetic modifications, age and mode of diagnosis, as well as environmental factors. Implementation of some of the new omic approaches may have the potential to improve our current understanding of the nature of this variability. An analysis of airway epithelial gene expression profiles complemented with GWAS data found that pathways relevant to airway mucosal host defense were significantly associated with CF lung disease severity. And this highlights the important role of heritable host defense variation in determining the pathophysiology of CF lung disease, and how it is important that we understand what is happening even at the gene level. So CFTR, mucus, infection, and inflammation are clearly interconnected in the pathogenesis of CF. Now, the exact relationship between each of these components is not clear, but each one is an important therapeutic consideration. Although the rate of functional decline or the severity of disease are variable, the development of lung disease is highly penetrant in CF patients. In young children with CF, structural lung disease was associated with the repeated detection of neutrophilic inflammation and infection in the lungs in the first 6 years of life. So it makes sense that prevention has the potential to reduce the long-term impact of CF lung disease for these children. However, we still have a lot of work to do to understand the early course of CF lung disease and influencing factors, including early treatment. Mucus is part of the host defense system within the lungs. It's an active viscoelastic matrix that consists of a complex interactome of mucins, enzymes, and host defense proteins. Mucus is produced at low levels in healthy airways. However, in CF, more mucins and an abnormal mucus composition were present in CF bowel fluid. Mucin levels also correlated with inflammatory markers and were already present in children with CF without structural lung disease or bacterial infection. And CF lung disease is worsened by bacterial infection superimposed on this muco-inflammatory state. What triggers this persistent muco-inflammatory state remains unclear, but it has been suggested that viral infections and maybe aspiration play a role. So it seems that chronic muco-inflammation is a critical factor resulting in structural damage, as aggressive bacterial eradication failed to prevent developing bronchiectasis in children with CF or in CF ferret animal model. However, in the CF pig, continuous antibiotics from birth improve CT imaging abnormalities and airway mucus accumulation, but not airway inflammation, air dropping, and submucosal gland plugging in 3-week-old pigs. So it appears that early and continuous antibiotics impact some, but not all aspects of CF lung disease development in the various models and also in CF patients. So mucus plug-in provides a nutrient-rich environment for bacteria, and along with impaired host defense mechanisms, permits a cycle of acute and chronic infection, inflammation, and lung damage. The development of CFTR modulators has significantly changed the treatment landscape of people with CF. However, modulator therapy is a lifelong treatment, and it's not able to correct the genetic defect and does not cure the disease. The data to date show a clear efficacy in improving lung function and nutritional status. However, the response to modulators is variable, and lung function still declines over time in the setting of established disease, albeit at a much slower rate. If in the future we see that CFTR modulators halt the progression of lung disease, for the youngest generations then with preserved lung function, these drugs may be all that's needed. But for now, modulators may not be enough to control infection and airway inflammation, especially over the long term. And these are the major life-limiting factors for people with CF. So the development of novel mucolytic, anti-inflammatory and anti-infective therapies is likely to remain important for both early and late-stage CF. Studies have shown that modulators reduce bacterial colonization within the first year of treatment, but some isolates may eventually rebound over longer periods. The effect of modulators on inflammation is unclear, as there's evidence of both reduction and no effect on biomarkers, including neutrophil elastase. Obviously, a lot of what we know about host defense in the CF airways comes from pre-modulator times. So we have a lot of work to do to understand what host defense looks like with modulator therapy. Some of the early evidence suggests that modulator therapy in CF patients can change immune cell function, and also understanding how the airway microbiome changes in response to modulators will play an essential role in developing strategies for managing airway infection. So as a CF researcher, I've spent a lot of time investigating the status and function of proteases and host defense proteins in the lung. And I'm going to talk a little more now specifically on how this imbalance contributes to impaired host defense. So proteases are involved in a range of processes within the lung, such as cell migration, extracellular matrix remodeling, mucus production, receptor activation, and inflammation, amongst others. Not surprisingly, the majority of focus to date has been on the serine protease neutrophil elastase, which is associated with bronchiectasis in children with CF. CF neutrophils release more elastase than controls, and levels increase significantly in patients chronically infected with Pseudomonas. This elevated activity then overwhelms the host's endogenous anti-elastase shield by cleaving some of its cognate inhibitors, such as SLP and melaphin. In vivo, knockdown of elastase reduced inflammation and mucus hypersecretion in the beta-ENaC transgenic mouse model of CF-like lung disease, demonstrating an important role for elastase in the pathogenesis of the muco-inflammatory phenotype. However, despite years of research, any inhibitors have had limited success in trials to date, and one of the reasons suggested for this is that a proportion of elastase remains bound to the surface of neutrophils, or it's packaged in extracellular vesicles, and these forms may be more difficult to inhibit than free elastase. So in the lab, we're particularly interested in the cysteine protease cathepsin S. Like elastase, it's a potent elastinolytic enzyme. So we identified CF airway epithelial cells as a source of cathepsin S and found that expression and secretion were increased by a dysregulated microRNA expression. Increased cathepsin S activity in the CF lung may also have a detrimental effect on host defense, as cathepsin S can cleave a number of antimicrobial proteins, including defensins, LL37 and lactoferrin, thereby modulating their activity. Like elastase, free and active cathepsin S was detectable in early CF. In fact, it was present in all CF bowel fluid samples that we analysed compared to elastase, which was detectable in approximately 40% of samples. Unlike elastase, cathepsin S activity was not infection-induced. And CAD-S activity, there was a negative association with lung function and a positive correlation with neutrophils in bowel fluid. So we then evaluated the role of cathepsin S in the pathogenesis of CF lung disease and its potential as a therapeutic target. To do this, we used the beta-ENaC transgenic mouse model, and we generated cathepsin S knockout beta-ENaCs, and we also used cathepsin S inhibitors. So both genetic and pharmacological knockdown of cathepsin S dampened inflammation and mucus obstruction when given either as a prophylactic treatment to newborn mice or as a therapeutic to adult mice. These effects were found to be mediated, at least in part, by the protease-activated receptor PAR2, which CAD-S has been shown to activate. So what does this all mean for the future? So I think basically we still have a lot of work to do and things to figure out. Why doesn't the CF airway innate host defense work? So I think what I've tried to do is highlight some of the reasons for this in my previous slides, but it's obviously a huge question. And we're beginning to understand more of the characteristics of CF airway epithelial and immune cells with the advent of new technologies, but more characterization is needed. New approaches enabling integrated studies of changes in gene expression, proteins, metabolome, Epigenetics, microbiome, and exposome are needed to capture the full picture realistically. Now, the most effective treatments may actually be those that target both sides of the inflammatory process. So working to enhance the pro-inflammatory response, working to reduce, the pro-inflammatory response, and also supporting the resolution of inflammation. And also targeted inflammation has to be balanced with the increased risk of infection. So maybe restoring defective components rather than interfering with a range of pro-inflammatory pathways might be a more successful strategy. And one area of potential interest within this field is the use of cell therapy. So of particular interest are mesenchymal stem cells, or MSCs, and these are a subset of non-hematopoietic adult stem cells with multilineage differentiation potential that originate from the mesoderm. MSCs exist in almost all tissues and can be easily isolated from bone marrow, adipose tissue, umbilical cord, and even the lung. And these cells then can be successfully expanded in vitro. MSCs and their secretome are multifunctional and exert reparative, antimicrobial, and immunomodulatory activities, making them an ideal therapeutic, at least in theory. So these activities have been demonstrated successfully in multiple animal and cell models of acute and chronic inflammation, infection, and lung disease. And they offer an attractive solution to the question of how to enhance the host immune response. For CF, lung disease, we're still at an early stage. Research to date suggests that we can learn a lot by capturing and exploiting the pro-resolution effects of MSCs. So in our lab, we've been looking at the effects of human bone marrow MSCs and their secretome on the lung phenotype in beta-ENaC transgenic mice. So we found that systemic delivery of MSCs exerted significant anti-inflammatory effects within the lung. Recruitment of mononuclear cells and neutrophils was reduced, and we also saw reductions in chemokines. We were particularly impressed by the significant improvement in lung function in adult mice. So in terms of a therapy, I think understanding the effects of these cells has the potential to provide us with new druggable targets for development. And hopefully I'll be back next year to tell you more about this story. So finally, I'm going to address some of the challenges I think we face in the future. With the increase in use of modulators, more patients are asymptomatic, which is obviously an amazing achievement, but it does create a challenge for researchers. Also, access to naive patients and naive samples will become more difficult. We don't know yet how modulators alter host defense mechanisms. And this is something we're going to look at as part of the Recover research program, which has been led by Professor Paul McNally and Professor Jane Davies. Then, if we do develop new treatments, will modulators alter their efficacy? When in the disease process should these new drugs be started? As early as possible would obviously be my answer, but this comes with its own challenges. We really need a better understanding of the pathology of early lung disease so that we can develop new effective preventable treatments. And then at the other end of the spectrum, we're seeing an increase in the population of adults living with CF. So what are the implications for long-term airway host defense? And what is that going to look like in these patients? So lastly, I'd just like to very gratefully acknowledge everyone in Belfast and beyond who has contributed physically, mentally, biologically, and financially, and emotionally to this work. I'd like to thank you all for listening, and I look forward to the discussion.
Okay, hello everyone. Thank you to our speakers. I'm Wanda O'Neill. I'm joined by my co-moderator, Deepika Polanini, and we will be moderating the question and answer session. Thank you for everyone who's entered questions. We've tried to keep up with them. I believe you can continue to add questions to the chat as we go along. So go ahead and continue to use that function in the Slido link. And so hopefully you can all see our panelists. We have all 4 of our speakers represented here on this panel. And I am going to just get started, sort of going in order of the questions that were received. We had some great questions. And so I will start out with a few questions to Kenichi. Kenichi talked about the small versus large airway expression of the CFTR and the mucin genes and how that can be integrated into a conceptual framework for how mucus moves through the lung, using some of the more modern technological advances, especially the single cell seq and the single cell in situ hybridization that Kenichi has worked out. So Kenichi, I hope you can hear me. I have a couple of questions for you. So first of all, let's see, which one should I ask first? So Kenichi, I know that, when you do your single cell RNA sequencing study and you look at expression levels, it's very helpful in determining, which cells might be functional. But can you help us understand, do we think that CFTR is important in alveolar type 2 cells, for example? I mean, we know that AT2 cells express a lot of CFTR, and yet, people don't talk about them in terms of function. So how do we relate these findings of where CFTR is expressed to like actual functional consequences of loss of CFTR in terms of disease development? Hope that question makes sense.
Yes, yeah. Thanks so much for, first of all, a lot of questions. So I'm trying to answer as much as I can. So yeah, the CFTR expression in AT2 cells are really consistently reported to be high, even sometimes higher than like airway epithelial cells. But we know little or no phenotype in the alveolar region in CF patients, and then I think that might be true because in contrast, all the ENAC channels are highly expressing AT1 cells, and then this is my personal speculation, but in alveolar regions, I think absorption may be dominant because the surface area covered by AT1 cells are much more than AT2 cells. And then it's important to absorb the fluid in the alveolar region because the gas exchange is most important function there. So I think that might suggest like CFTR is expressed, but ENAC and absorptive function overcome the expression. So that's why I think if CFTR protein is abnormal in AT2 cells, but still we don't see phenotype. So that's my speculation, but I basically agree. So that's really a largely unknown area. So, and then I think important to explore.
Yeah, so it brings up this concept, and the other panelists should please feel free to jump in at any moment. This is an open Q&A for us. So it brings up the concept of, CFTR is expressed, but it's also expressed in the context of everything else that's in the cell, other channels, other types of channel regulators. And Kenichi, how much do you think, and others, like how much do you think it matters that we explore as we're thinking about lung physiology, that we explore the relationship of CFTR to all of these other channels, for example, all the ones that are considered to be alternative treatments for CF? Maybe we can open that a little bit.
Yeah, I think that's very important. I think probably the two things we should consider to relate CFTR to the other channels. So one is like cell type, because like in disease, cell type, like cell proportion may be altered in the disease inflamed environment. So that also make change in the like total expression level of those alternative channels. For example, TMEN16A, which is AN1, is highly expressing goblet cells. And in small airways, as I showed in my presentation, the goblet cell population is much less in small airways. So that also make the difference in those alternative channel expression depending on region. So yeah, and also so that, and also what I know about alternative channel expression in regional difference is I know like one paper published describing ATP12A, the proton pump channel is highly expressed in large airways, and then even different type of proton pump works in the small airways, in at least peak airways. So, and I looked at my own data sets, like single cell RNA sequence data set from large airway and small airways from human tissues, and then so far the beta ENaC is not much significantly different, but some subset like alpha and beta tend to be higher in small airways. And then KB channel like KCNMA1 is almost no difference in large and small. And the N1 is no difference. So that's what I know, but I have not systematically looked at those alternative channel expression in different regions. So I should explore those expressions.
Dr. Salafi, would you have any thoughts related to that issue and how critical it is to consider these channel expression in the context of where CFTR is?
So I think it really depends on which channel we are talking about. You know, some of them might be independent, some of them might be dependent. And then it's questioning, when do you intervene with modulators and how do you then influence the whole disease? I think there are really differences between early disease and then later disease and how you connect the two. So for instance, you know, what I said, the SLC26A9, there are variants that are seemingly better, even making Cftr treatment of the G551D mutation. So there is a link between CFTR and SLC at 26A9. So there is clearly an important piece. Whether TMEN16A is important or not, that may depend on the expression that Kenichi said also in goblet cells and when it's expressed. And, you know, this might be upregulated and then more amenable to therapy. However, if you look at the sheep data, at least, the TMEN16A seems to work even without stimulation of inflammation in the peripheral airways, at least according to the mucociliary function. So I just don't know whether we know enough about this, and when we really need to look at that. Again, it's likely very different if you start modulator therapies very early on, which we cannot do yet, but very early on, maybe even in utero, versus starting therapy with any of these modulators of any of these channels later when actually airway disease is established.
Yes, very good. I think you're right on there. Okay, so let's move on to some questions that relate to Dr. Quinton's talk. Dr. Quinton, thank you for joining us. And I think everyone found this concept of the luminal fold and the basal plates to be very interesting. And there were some questions about whether or not there is, Kenichi, this also would relate to you, whether or not we have evidence from a single cell level of specific expression of cell type differences that would relate to the pleat and the fold. Maybe we could start with that question.
Yes, yeah, if I can speak first, yeah, about the single cell part. So yeah, I would say it might be challenging to identify the specific secretory subset specifically localized to the pleat or fold region, because we usually lose spatial information in the single cell sequence set. But if we have like known specific markers, so we can probably just focusing on those identification of those secretory subsets in the single cell data set.
Well, I mean, I think it is interesting. We have these distributions of cell types from top to bottom, and then even at a single spot within that distribution, we seem to have another distribution of cell types based on Dr. Quinton's work. So maybe just by knowing that, we would be able to sort of tease that out. Dr. Quinton, is there any correlation or ability to integrate what you see in these folds and pleats to what goes on in the intestine related to CFTR?
I'm sorry, I didn't quite understand that.
Well, there's villi and crypts in the intestine. I think the question is related to, do you sort of see the same kind of CFTR expression in the intestine that you're seeing in the folds and crypts of the airway?
I honestly don't know how CFTR is distributed in the intestine. So, but we did take our inspiration for developing the folds and pleats model from the intestine. And, in both cases, we have secretion and absorption, which are in completely different cells, and they're going on simultaneously.
Wanda, if I may interject, it's a great question because I know that there has previously been shown localization of NF-kappa B signaling in crypts and denuded lamina during intestinal infection with specific bacteria. So the regional activity of CFTR in the crypts and villi in the intestine would also be of, I think, significant interest in that intersection.
Yeah.
Dr. Quinton, do you know how far down these pleats and folds go? Even within the bronchioles now at a molecular level, we're getting to the point where we have respiratory bronchial molecular signatures, terminal bronchial molecular signatures, and then, the larger bronchi, and they have a different signature. So it seems like it would matter in terms of how these pleats and folds work to know how far down they go. Do you have any idea?
Well, I should know the answer to that exactly, but I don't. In our own work, we've dissected the small bronchioles, but not respiratory bronchioles. So my guess is that CFTR is prevalent in all of those because we begin with pathology in this very small bronchioles. So, and those are the most vulnerable to infection and I guess damage in the beginning. So I'm not very satisfied with my answer, but anyway.
Yeah, I mean, like when Kenichi looks in his single cell data, I think it might matter where he looks. That's my point. Like if the pleats and folds kind of morphology is not present in the very terminal bronchioles, maybe you wouldn't be able to find the differential cells. But if you go a little higher and that exists, then that's maybe where you should look for them. That was sort of my point there. And we had another question from one of our listeners, Dr. Quinton. The question is related to whether or not you think that gene therapy will be affected by this sort of morphology. That is, do you think that those cells in the fold are going to be more likely to be more difficult to transfect and thus more likely to be difficult to correct?
Well, it seems logical from sort of three-dimensional vision that they would be, but I'm not all sure of that either. And part of the reason I would say that I wouldn't be sure of that is because when we are breathing in and out, we're stirring the surface of the epithelia because the pleats and the folds are expanding and collapsing. So I referred to that in my article on this as a washing machine. And so if it's acting like a washing machine, I think that we're going to get exposed in the pleats as well as the folds equally, or will.
That's a good point. Okay, thank you. Let me look at my list of questions. Okay, I have a question for I'm sorry, I have to scroll through my chat to get to the questions. And it's awkward. Okay, this question is for Dr. Salate. It's related to losartan use. Can you talk about losartan use in people with CF? How much is it already being used? And what are the other clinical implications or risks for patients on losartan?
So losartan is actually not very commonly used in cystic fibrosis at all. Some patients with diabetes may be on it, but there are very few when we looked at the CF registry. But the angiotensin receptor blockade, which doesn't seem to be the major activity of losartan in this case, but more its anti-inflammatory activities. The medication is extremely safe and has been shown to be well tolerated with very few side effects. Even when people think that it lowers blood pressure, it seems to do that only in patients with high blood pressures, and then it levels out at normal blood pressures. So side effects from all these could be worsening renal function, but it's actually used to protect the renal function in diabetes from getting worse. So the side effects are rare, sometimes muscular, but this is a medication that could be widely used if shown to be effective. Let's put it this way. We haven't really shown in human beings that it's effective, in CF in particular.
And there is another question for you. So it was interesting that the glucose concentration affects the BK channel activity. Do you know if we could expect it to also affect other channels relative to CFTR?
So you know that in normal cells, it's actually increasing CFTR activity, which is interesting. And therefore, in normal cells, there is sort of a yin-yang between CFTR and the other channels, not affecting it as much unless you take CFTR function down. Now, in patients with cystic fibrosis, CFTR is not fully functional. And even if you increase the CFTR function with now highly effective modulators, the BK channel decrease is still causing mucociliary dehydration and therefore mucus hyperconcentration. So it affects other channels, sometimes in better ways, but the detrimental effects predominate specifically in cystic fibrosis. So there we need to do something, hopefully, that we can balance that out.
Very good, thank you. So I have a question for Dr. Walden. Do you think that correcting the CFTR in airway epithelial will be sufficient to cure the CF lung? So there's lots of interactions, obviously, once disease develops. I know you called it the million-dollar question in the chat, but we would like to get your thoughts about that question.
Yeah, I think it's a difficult question to answer, because obviously a lot of the recent work suggests that given patient modulators does alter immune cell function, but we don't know yet whether that's as a result of an interplay between the epithelial cells and the immune cells, or whether the modulators are actually able to directly affect the immune cell function. So I think it might help, but I suppose I'm a little bit kind of disheartened, I suppose, by some of the recent results, you know, that we're not seeing sustained effects on inflammation and infection in patients with modulators. Obviously, we're still, you know, it's very early on. We still have to see what happens more in the longer term, and obviously with the newer treatments. So I don't think it would be sufficient. And I suppose part of that is, why is it not sufficient? You know, what else is going on there? So yeah, I think a lot of stuff to figure out we have to do in the future.
I mean, if the modulators aren't, this is a question I just thought, if the modulators aren't working on inflammation and immune cell function, why would that be? I mean, it's a systemic treatment. So would there be some reason you could imagine why CFTR is not corrected in those cells?
I don't think so. I'm not off the top of my head, you know, because we're seeing improvements in lung function, you know, obviously the systemic treatment is working in the lungs. So why we're not seeing associated changes in inflammation, I can't answer that yet.
Right.
But yeah.
Very good.
Perhaps one of the caveats to that is that if we have effective delivery in the airway for gene therapy is upcoming, then, you know, is that in itself a model where we can see what the sole effect in the airway epithelia are relative to the contribution of inflammatory cell correction?
Yeah, because I have to assume that the modulators obviously have the potential to alter immune cell function. They're not targeting epithelial cells specifically. So it's a possibility, just I think we still have a lot more to figure out in terms of mechanisms.
Dr. Weldon, we also have a question from Tracy Bonfield for you. So she was interested in your HMSC studies and wonders, did you see improvement in ion transport in addition to lung function and anti-inflammatory effects? And she wants to know if you utilized autologous mouse HMSCs in your model.
So unfortunately, we didn't look at ion transport in the BD-NAC model. And we just used human bone marrow derived MSCs, partly because just trying to keep the clinical relevance. But I suppose one of the reasons why we tried human MSCs in the BD-NAC model is because the lung damage that we see in that model is just notoriously resistant to therapeutic intervention. You know, a lot of times when we see a reduction in the lung damage, it's because of, you know, sustained knockdown from birth, but the MSCs actually worked as a therapeutic. So giving them to adult mice was able to restore lung function. So, and obviously we see the knockdown then in inflammation and inflammatory cell recruitment, but I agree, like it'd be very interesting to look at ion channel activity in cells from the mice as well. Yeah.
So I have a question related to that, is how do you envision the HMSCs like moving into human? How would that actually work in your mind?
In my mind, and this might be, I don't know, it might be controversial. In my mind, I don't know about chronic disease. You know, I can understand them, you know, in terms of an acute inflammatory response or acute condition, but as a treatment for chronic disease, I think we have a lot to learn from these cells. You know, they're, I say, like they're so potent, and the fact that you can see effects, you know, 2 weeks after administration, they're just remarkable. And what I would hope is to learn more from their mechanisms of action to try to develop, you know, other druggable targets. And I think developing MSCs as a therapy for CF, I think it's any chronic condition. I can't envisage that in my head at the minute, but.
Well, let me just give you, I'm just going to give you a little encouragement. I mean, from my own experience, this is not with CF, but my son had a fistula. He has Crohn's and had a fistula, and as part of an experimental treatment, they, I don't know if he was on placebo or whatever, but the MSCs were an experimental clinical procedure or in a trial to try to close the fistula. And whatever they did worked. So I think whatever it was they did, whether it was the MSCs or not, I just encourage you to continue to be excited about that cell type and what it can do for inflammation.
Yeah, exactly. I think we have a lot to learn from them. They really are fascinating.
Okay. Let's see, I have some new questions. So Kenichi, there's a question for you about the functional correction of CFTR and using the CCSP promoter in the rat, do you know yet, are you achieving similar levels of CFTR with that promoter? Do you think there might be some issues related to promoter activation that might be causing you troubles in your studies?
Yes, that's a really important question. Yeah, we have not compared like promoter activity of our CCSP, rat CCSP promoter versus the original CFTR promoter. So, and also we have not compared the CFTR protein levels in those like our rat CCSP CFTR transduced. Probably cell type is better, so like secretory cell versus non-CF secretory cells. But now we have cultures from large and both small and large airways. I think, and also we have identified some small airways secretory cell markers. So I think we can sort secretory cells and then directly compare mRNA and the protein in non-CF versus like those transduced cells.
Okay, thank you. Dr. Slafi, there's a question from Katie Hissert. She's in the Fellows Adult Clinical Cases Unit, and she reports that there were 2 cases of myocardial infarctions yesterday. And she says, as our patients age, we may start using more ACE1 and ARBs in our adult patients. Do you have any thoughts about initiating these earlier?
So there's my bias, right? I think we should start very early, but I believe that also we need to show some positive effects first. I don't, if they really truly have anti-inflammatory effects, like other medications that are much more toxic and not well tolerated, these are well tolerated and they have positive effects. They have been shown in other diseases, like I said, in diseases, including cardiovascular diseases. A lot of the effects are not necessarily only related to the antihypertensive effects, but they're actually related to their anti-inflammatory effects. So they could subdue some of the inflammatory effects that we see in CF as well. So I'm a big fan of them. So I would start them early, but I think we need data. We need objective data to show that they are actually doing something positive. They're not really used otherwise for prevention of primary events. So we would have to show first that we do something meaningful.
Okay, thank you, Katie, for your question. Dr. Quinton, there's a question for you from Jonathan Zuckerman. He asks, have you thought about the physiological effect of the equal pressure point, which migrates along the airway during forced exhalation? How do you think migrating airway collapse during forced exhalation integrates with your cross-sectional model of the small airways in CF? I hope you understood that. Dr. Quinton, are you still there?
It looks like we may have dropped connection with Dr. Quinton.
Oh, I'm sorry.
No, I just noticed that myself, but we do have a number of questions that have received upvoting. So if we go to the top of our screen, those are prioritized there as well.
Okay, DB, help me out. If you can see them and want to ask them, go ahead. I don't have a problem with that.
We have a question from Clement Brito for Dr. Weldon. And Clement says, wonderful talk, Sinead. Do you think epithelial CFTR correction would impact airway immune cell subpopulation development and maturation? For example, CF-specific airway recruited monocyte and macrophage subsets. I guess this is somewhat a follow-on to the previous discussion, but perhaps we can expand a little there.
Yeah, I think, as I said before, the literature obviously suggests that modulator therapy has the potential to alter immune cell function, mostly looked at neutrophils. But I suppose like all this new findings, that we previously thought that neutrophils were, terminally differentiated, but this idea now that they can be modified transcriptionally and phenotypically by their environment, you know, in response to, say, transmigrating through, I think the experiment was through CF airway surface liquid. You know, if we put CF airway surface liquid from patients with modulators, what would we see, you know, in terms of the response from the neutrophils? So yeah, I think we have a lot to learn, because obviously you have the relationship between the airway epithelial cells interacting with the immune cells, but we also have CFTR in the immune cells. So how is CFTR correction going to alter those? We still have a bit to figure out.
Thank you. Kenichi, there's another question for you. I know it's a difficult question, but it's from Emanuela, and she asks, how does expression of CFTR correlate with protein levels? So ciliated cells express low levels of CFTR mRNA, and we know that immunofluorescence data, at least in the past, has shown that the CFTR in ciliated cells is actually quite substantial. So what is the level of CFTR protein in secretory cells versus cilial epithelial cells, and how would you get at that question?
Yes, yeah, we have performed like immunohistochemistry using previously published antibodies, and then we could identify ion sites always, because I think the CFTR protein expression is very high compared to other cell types. That's, I think, consistent with single cell RNA sequence data, but vary in the apical membrane in the secretory cells, and then almost really rarely or rarely little in the ciliated cells. So if we use ion site as an internal positive control, so and then we think the antibody works, so the protein level expression of CFTR in ciliated cells are also, I think, really not so high based on our immunohistochemistry, but it could be an antibody sensitivity issue. So yeah, that's one issue. Like antibody sensitivity is one issue. So yeah, for low abundant protein level. So, but sorry, I cannot answer the good question. I don't have that good answer, but yeah, I wish we had like single cell proteomics or those kind of new technology to identify protein levels per cell type.
It's obviously a very good question, and you know, it will be very important to develop the kind of assays that we need to know whether or not CFTR, even though it's not expressed at very high levels, whether or not it's actually functional in ciliated cells.
Yes,
I know
also
that a lot of people are working on that. Go ahead.
yes, yes. Also, there is transitional cell population probably from basal to secretory or secretory to ciliated, and then we don't know which level of transitional cell type express CFTR, and then from which level, so we lose CFTR over the differentiation to other cell type. So those are also, I think, important questions. Yeah.
Okay, Deepika, how are we doing? Do we have new questions?
Sure. We still have about 13 to 15 minutes in our session. So I think the next question that has received quite a bit of likes or moving up to the top is also going back to Dr. Salate. And this is, again, it's kind of a follow-on of some of the discussion that we've had, but how much do we know about the expression of the channels that you discussed in the regions of the folded small airways that Paul discussed? And this has been somewhat answered, but maybe you want to expound.
Well, I can't really expand on that because, you know, we don't know the exact locations of these channels short of the single cell RNA-seq data. And what Kenichi said in terms of distal versus proximal. But the exact locations we don't know, or let me put it this way, I don't know, maybe some people do know. I think folding is interesting in terms of ways of how to signal locally and do this. This was actually a concept a long time ago thought by, I think Jeff Drazen was in this, where in terms of asthmatics with smaller airways, and then expirations as well. And that has potential effects on the paracrine signaling as well. So it could be relevant there significantly. There was one question about equal pressure point. Obviously, that is only happening during forced expiration, and that could trigger these signals as well. But where they exactly expressed and what they do, I don't know.
So that sounds like more mining from your data, Dr. Okuda.
We have a question. I'm sorry, DB, we can take turns here. We have a question for Dr. Weldon. Do you think the CF inflammation is associated with accelerated lung aging? I think that's a very interesting question. Any thoughts about that?
Yeah, like there are some reports of senescence markers, and obviously the SAS phenotype in, the pro-inflammatory response in CF is similar to a lot of the SAS phenotype. So yeah, like there could be a relationship there. Yeah.
Okay, go ahead, DP.
Oh, I was just going to say that we have several comments from our colleagues in Ohio. So from Tracy Bonfield, a note that they have done the first in CF human mesenchymal stem cell clinical trial in Cleveland with a manuscript in preparation, which will be certainly interesting for future discussion. So that was a share out for the audience. And Ben Kopp shared asked really, did you do immune cell responses to modulators showing improved outcomes? And do you wonder if that will work in the lung? So some additional questions that way. I think one of the questions from Dr. Ribeiro at UNC could probably come up next. Wouldn't the lack of an anti-inflammatory effect by CFTR modulators indicate that defective CFTR is not directly implicated in inflammatory responses? For example, these findings go against the notion that the intrinsic CF defect directly causes hyperinflammation. So I think this is a bit of a back and forth debate with also intersection based on the data that you showed, Dr. Salate, of going back to alternative ion channels and impacts from inflammatory stimuli there. So perhaps Dr. Salate and Dr. Weldon could engage in answering on this question of intersections of inflammation, alternative ion channel transport, and modulator function, which really brings in almost all pieces of our presentations today.
Yes, and DB, it links in with another comment by Katie Hissert, who says, can we learn lessons from those that don't have CF, for example, non-CF bronchiectasis? So her point is that if you have established lung disease, maybe curing the CFTR defect in the epithelium is not going to lead to like a complete correction of problems. I mean, we have people who, non-CF bronchiectasis is sort of a syndrome that's like modulated or corrected CF people. They get bronchiectasis in the presence of normal CFTR. So what is the pathogenesis that causes that kind of pathways? And one of the things that we've been thinking about here, and I think Umakami has a poster on it. Unfortunately, I don't have that up exactly right now where that is, but whether or not it might be related to chronic hypoxia and the feedback cycles that relate to chronic hypoxia in diseases such as non-CF bronchiectasis. So a very good point. And, getting back to when do you start these modulator therapies? I think everyone thinks if it's safe, the earlier the better. Try to prevent the structural lung damage and try to make the modulators even more effective long-term.
So I think we could probably look to both Dr. Weldon and Dr. Salate for their comments on that kind of complex intersection and using both CF and non-CF bronchiectasis as models or as a kind of background model. And we'll start with ladies first with Dr. Weldon.
Like I really like that analogy of, like CF patients on modulators are similar then to non-CF bronchiectasis. That's, yeah, it's definitely a tough one. In terms of the whole, like, it's the chicken and egg kind of scenario of, is it defective Cftr that causes infection and inflammation, and which comes first and that. But I think like a lot of the studies now on early CF and kids with CF showing like the structural changes and the presence of altered mucus composition and these kind of things being detected very early in the disease pathogenesis suggests that there is a link. But in terms of why the modulators aren't sufficient to correct the inflammation, I don't know whether it's just that we haven't achieved the maximal threshold for affecting immune cell populations. I don't know. Yeah.
Any other comments on this issue from the panel?
I just wanted to say, it always depends what you define. What is a normal ion channel function? And what ion channels actually need to function at what level to keep the airway whole? And in non-CF bronchiectasis, yes, there is no genetic defect in cystic fibrosis transmembrane conductance regulator, but over time, an insult caused something that is also associated with an abnormal mucociliary function, at least in the areas that are affected. So whatever sets this off is then responsible for maintaining it. And I believe, again, what you said, Wanda, when you start early, you may be preventing it. If you don't start early and you have the effects already, it is very difficult to reverse them. You may be able to keep it going at a reasonable level, but to reverse it, I think that's the real question, what you need for that. And CFTR function alone may not be sufficient, especially if you still have some of the inflammation that was talked about that affects other channels as well.
Yeah, I think, like that, because we're given modulators, obviously at a more advanced stage of disease, we haven't fully characterized yet, epigenetic changes, given the modulators like that will just not be enough to restore everything back to normal because of what the body has been exposed to in the years before. Whereas obviously if we were able to start effective treatments as early as possible, you're limiting that progression. But I think going in at, 17, 18 years of age, we're just not going to be able to correct everything because the damage has already been done and just things have changed that modulators will not be able to reverse.
And we know that not all patients respond the same way to modulators. So, I mean, there are many more things going on than just a simple modulator, simple in quotation mark, that corrects CFTR.
Yeah.
So we had a question for Dr. Quinton. I know that he's not on, but I think maybe some of our other expertise on the panel could answer the question, or at least help the group think about it. And the question is, if we have secretory cell hyperplasia or goblet cell metaplasia, and the concept of this washing machine concept, do you think that would affect that? And my thought would be that it would, the way that Dr. Quinton described it as fluid secreting versus fluid absorbing, secretion in the crypt and absorption in at the tip in the lumen, that anything that disrupted the ability of the channels to work properly in that kind of morphology that he describes would affect that ability of the function of that fluid moving to work. So that's just me talking off the top of my head, and maybe others on the panel would have some thoughts to help answer that interesting question. So the point is that it might matter in asthma as well, right? It might matter in COPD, not just CF, where these absorptive and secretory cells are located. And if they change to a goblet cell and they don't absorb anymore or secrete properly anymore, it seems like that would matter. I agree. Kenichi or?
Yeah, I think that's a good point. And I also wonder, like, if this is kind of chicken and egg, but if goblet cell metaplasia tend to be easily happen in portion of the small airway epithelium, so and then that could make like fold or like fold first. I'm not sure, but as you mentioned, like in asthma, so there is goblet cell metaplasia all over the epithelium. So yeah, I think that concept may be altered, and then that could worsen the disease. So yeah, that's my question.
Okay, very good. I have been informed in my little chat box that we are at time, and if anyone has any concluding remarks that they would like to make, thank you, everyone. We didn't get to every single question, but I think we tried to get through most of them. I'm sure the speakers would be willing to answer individual questions about their sessions if you reached out directly to them, or feel free to contact me or Deepika as well if you would like us to reach out to the speakers on your behalf. So Deepi, any final comments from you?
I just want to thank all of our speakers and really say this has been terrific engagement. We had more than 370 participants watching the session today, over 30 questions, I believe. And I just really want to thank everybody in the audience. I know that we all would prefer to be together in person for NACFC, so thank you for joining today. And please do email our speakers directly if there's anything that we weren't able to touch on. Thank you, Wanda.
Thank you, everyone. Have a good rest of the CF meeting. We hope it's very productive for you. Thanks for putting up with me.
Thank you.
Thank you.
All right. Bye-bye, everyone.